was haploid production, and then definitely doubled haploid production in plants, particularly in wheat. So if we talk about this, we in the class on Thursday, we discussed that in plants there are two methods for haploid production, double haploid production. First is microspore culture or anther culture, pollen culture or anther culture. So here what you can see is these are wheat plants. In the figure A, you can see that here they have placed the plants at 4 degrees centigrade. What does it do? It will increase, it will enhance the pollen efficiency, pollen's efficiency to grow on growth media. So for 24 hours, the plants are placed at 4 degrees centigrade. And the figure B, you can see that here we have successfully isolated the anthers from the wheat plants. So these anthers are shifted into a petri dish and they are sterilized. Okay, here you can say we have kind of, in the figure B, you can say we have kind of washed them to remove any debris, etc., present on their surface. In the figure C, you can see that we have shifted them into petri plate, and now we have to sterile them. We have to make them sterile. So usually we use 0.5% to 1% bleach for sterilization of for the sterilization of explant. So we will use bleach solution to make these anthers sterile. Then these anthers are shifted onto MS media or N6 media, which is also called as chew media, and it will contain usually 2,4-D hormone. 2,4-D is an auxin that is used in 2,4-D is an auxin that is used for the production of callus, or it is used to grow the anthers into whole plants. So usually what will happen after some time, these anthers will grow into the callus, and then callus can be subcultured, and finally it will be shifted on to rooting media and shooting media. In this way, we will get haploid plants. So in the figure E, you can see that from the callus, now we have plantlets. Plantlets means small plants. So they have roots and shoots. Then these plants are shifted into peat and soil mixture. I have already told you what is this in previous classes. So in this mixture, we provide nutrients to the plants and the plants will grow. Once the plants are like almost 1.5 to 2 feet in height, then we will do colchicine treatment. What we will do? Colchicine treatment. For the colchicine treatment, we cut the roots of the plants except 1 inch. So if the root length is like 5 inch, we will cut the lower 4 inches of the root. Only 1 inch of the root, 1 inch of the root will remain. What we will do is this solution has colchicine, and it is usually 0.2% to 0.5%. So it means that if you are preparing 100 ml of solution, you will add 0.2 gram of colchicine. If you are producing 0.5% of the solution, you will only take 0.5 gram of the colchicine and dissolve it in 100 ml of water. So in this way, the chromosome number will double. I have already told you in the class how it will double. The colchicine breaks the spindle fibers of the chromosomes during the cell division. So the chromosomes will not move to the opposite poles, but they will be restricted into a single cell. In this way, the number of chromosomes in the plants will double. And after that, we will again shift them to the pots as shown in figure A. And here we will put them in controlled condition. In the in greenhouse, we will make sure that light, temperature, humidity, etc are maintained. We have already discussed in our previous classes that relative humidity should be around 60 to 90% for most of the plants, and the temperature should be around 22 to 25, 27 degrees centigrade for most of the plants, because it is wheat, it is a winter plant. So 22 to 25 degrees centigrade is a good temperature. Even 22 is an ideal temperature for wheat. So in this way, the plants will regenerate the roots that we have cut in the figure A. And once the plants establish themselves, we will shift them to the field for seed production as shown in figure H. So this is one way that we discuss on the class, on your class on Thursday. So this is one way. The other way is there are only two types of cells in plants that are haploid in nature. Number one is pollen grains and number two is egg. As we have already discussed in the class, that eggs are very small, number one, just like pollens. They are even smaller, and but they are protected in a complex structure called ovary, ovules, and ovary, then ovules, etc., etc. So they are very fragile in a sense that if you dissect the ovary, if you dissect the ovule, the eggs will be damaged. So there is an option that instead of isolating eggs for the production of haploids in plants, what we can do is we can just easily manipulate its reproduction system. How we can manipulate its reproduction system is that the process is called wide hybridization. The process is called
Wide hybridization.
wide hybridization. So the second method for the haploid production in plants is wide hybridization. What is wide hybridization? So if we cross wheat with wheat, it will be called hybridization. If we cross maize with maize, it will be called hybridization. If we cross rice with rice, it will be called hybridization. But if we cross sorghum with sorghum, it will be called wide hybridization. But if we cross wheat with maize, that is another species, it will be called as wide hybridization. If wheat is crossed with sorghum, it will be called wide hybridization. If wheat is crossed with barley, it will be called wide hybridization. Wide means Urdu mein choda, not narrow but wide. So this is called wide hybridization. So the most common method to produce haploids in wheat is crossing wheat with maize. Wheat into maize wide hybridization or wheat into maize wide cross method is used for haploid induction. So there are some alternative methods as well. Number one, we can, instead of this wide hybridization, maybe we can use wheat's pollens, but that pollens will be irradiated with the X-rays or gamma rays, so they will become sterile. So still they are wheat's pollen, But once they are crossed with wheat, they have already lost the ability for fertilization. So there will be pollination, but there will be no fertilization. What does it mean? That because wheat's pollen have been irradiated with X-rays or gamma rays, they have made them infertile. Once they cross with wheat, they will send the signal that the cross has happened, but there will be no pairing of the chromosomes from the anther pollen because they have been irradiated. These pollens have been made infertile. So the resultant embryo, what are we talking about? Not the egg now, but we are talking about the, what we are talking about? We are talking about embryo.
Embryo. Oh,
Embryo. So wheat's egg is crossed with wheat's pollen, but that pollen is irradiated. So it will do the pollination, but these chromosomes are infertile. You can say that these pollens have chromosomes that will not pair with the 21 chromosomes present in the egg. So there are 42 chromosomes in wheat. How many chromosomes? 42. In a diploid wheat plant, there are 42 wheat chromosomes, but in a haploid one, there are 21. So in pollen grains, there are 21 chromosomes. In an egg of wheat, there are 21 chromosomes. So if we cross wheat's egg with a pollen which has 21 chromosomes, but they have been irradiated with gamma rays or X-rays. So the 21 chromosomes from the pollen grains will not pair along the 21 chromosomes from the egg. So what will happen? What will happen is that there will be no pairing and the resultant embryo, first when the egg and pollen grains are crossed, we will have zygote, and after several uh, mitosis events, after several cell divisions, we will have, we shall have an embryo. So embryo has, pollen has one cell, egg has one cell, zygote is still a one cell, but once it undergoes several cell division or several mitosis cell division, it results into embryo, and embryo is multicellular. It has hundreds of cells. So this embryo can be cultured on the growth media, and this embryo will give us haploid plants. Same is true with wheat into maize cross. So wheat has 21 chromosomes in its egg. Wheat has 42 chromosomes in diploid form, but because egg is haploid, it has 21 chromosomes. So the maize is an other cereal plant that has 20 chromosomes in diploid form, but once we get its pollen grains, we have only 10 chromosomes in its pollen grains. So what we do is we emasculate the wheat. We have already discussed how to emasculate the wheat flour, the wheat spikelets. So what we do is we cut the upper part of the wheats spikelets, and we, by using a forceps, we carefully remove the anthers from the wheat spikelets. Only ovary or carpel is left. So that carpel will be crossed with maize chromosomes, I'm sorry, maize pollens. So when The zygote is formed. When the zygote is formed, the zygote will have 21 chromosomes from the wheat and 10 chromosomes from the maize. You can easily understand there will be no pairing. So the genetic material, the genetic stuff, the types of chromosomes are different. Number one, their number is different. In wheats, from the wheats, eggs, 21 chromosomes are coming. From the maize pollens, 10 chromosomes are coming. So pairing is impossible. Number two, they are not homologous chromosomes to each other. They are not identical chromosomes. The chromosomes that will come from wheat's pollen will be same like the 21 chromosomes coming from the wheat's egg. But these are two different species. So their size will be different. The genes present on these chromosomes will be different. So they will not pair. Number one, they are not homolog. Their number is different. Number three, the genes present on them are different. So number four, the kinetochore region. What we are talking about? The kinetochore region. What is kinetochore region? Kinetochore region or centromere is a region in the center of a chromosome where spindle fiber attach. So it's wheat's egg and the spindle fibers of our wheat, so they will not attach to the centromere or kinetochore proteins present in the centromeres of the maize chromosomes, so they will be lost. This is called chromosome elimination. But the signal was sent that pollination has started, so the zygote will keep dividing, and it will result in a embryo. But that embryo will not be a true wheat embryo. How? Let's restart it again. For example, these are maize plants in the figure A. So we will collect pollen grains from them. On the in the figure B, there are weeds spikes, and then you can see that here spikelet 1, spikelet 2, spikelet 3, spikelet 4, 5, 6, 7, 8, 9. So in each spike, there are a lot of, usually 8, 9 or 10 spikelets. So as we have already discussed that we will cut the upper part of these wheat spikelets, and then we will take pollens from the figure A, or you can say maize plants, and we will sprinkle them, we will spray them on these emasculated wheat spikes. So I have already told you that these wheat spikelets have been emasculated. Emasculation means that the male flowering part has been removed. First, we will cut the upper part of these spikelets. As you can clearly see that they have been cut, and then the we will carefully remove the, can anyone tell? Anthers. We will carefully remove the male part of the flower, anther, from these spikelets, and only carpels will be left, means only ovary stigma style or carpel will be left. So once we sprinkle these pollens from the maize onto the weeds male flowering parts, sorry, weeds female flowering parts, of the a signal will be sent to the ovules that pollination has happened. The pollen will travel through the pollen tube and it will reach the ovule. It will cross the weed's egg. So I have already told you in weed's egg there are 21 chromosome haploid number, and in maize there are 10 chromosomes. So 10 chromosomes and 21 chromosomes will not bear, number one. Number two, their lengths, chromosomal lengths are different. So number three, because they are not homologous chromosomes, the genes present on wheat and maize chromosomes are different. So these maize chromosomes, in their centromeres, there are kinetochores that are specific to the spindle fibers of the maize and not to the wheat. So no spindle fibers will be attached to them during cell divisions. So after few cycles of the mitosis, not immediate, after few cycles, when zygote is being converted into embryo, these maize 10 chromosomes will be lost because they are not being attached to the spindle fibers. So they will not attach to spindle fibers. So they will not stay in any cells during the cell divisions. So there is a defense mechanism in plants. Any foreign particle like bacteria, like enzymes, so they are immediately killed. So they will be recognized as foreign invaders. So these chromosomes will be destroyed. These chromosomes will be lost. This process is called as chromosome elimination. What did I say?
ये प्लांट्स जो हैं वो इनको जो दूसरे बाइट्स हम लोगों ने पोलन्स डाले थे उनके जो क्रोमोसोम्स होंगे उनको फौरन कंसिडर करेंगे एज ए फॉरेन पार्टिकल्स और उनको एलिमिनेट कर देंगे इन अ प्रोसेस ऑफ क्रोमोसोम एलिमिनेशन.
So the chromosome elimination is a slow process. Definitely, it is done in 2 to 3 or 4, 5 cell divisions, but definitely they will be lost. They will not attach to any spindle fibers. So, but the process has started. Zygote will be converted into embryo, but because there is a problem, there is half number of chromosomes, not the total ones. So the embryo that will be produced will not be a true embryo. The embryo that will be produced during this process will be an immature embryo. What will be it? An immature embryo. So in the figure C, you can see that after 2 to 3 weeks, we will have wheat seeds. So these green ones are wheat seeds that has formed in these spikelets that were pollinated with maize ones. So instead of having starches inside of it, it has a white fluid inside of it. So this is kind of an abnormal wheat seed. So this wheat seed is still green. Definitely, it will grow into yellow when it matures, but right now it is green. Another thing is because it is not a normal wheat embryo, so other processes will also be. So there is a white liquid you can see here, and this white stuff. This is immature embryo. What is this? This is embryo. So what we do is, definitely this is not as exposed at it. This sweet seed is closed. We cannot see the embryo, but by using a blade inside a laminar flow hood, we will have a gentle cut on the wheat seed. So then by using the forceps, we will open it. Once we will open it, we can see the embryo, and by using the forceps, we can collect this embryo and transfer it to the growth, transfer it to the growth media, transfer it to a growth media.
Growth media.
So there you can see that number one, on these spikelets in the figure B, the seeds will develop maybe in 3 to 4 weeks, and these seeds will be green in color. We will not be able to see the embryo. So we will do a procedure that is called as embryo rescue. What is it called? इसका नाम बताएं. What is its name? Embryo rescue.
Embryo rescue.
Number one, you did emasculation. Number two, you did pollination. Number three, chromosomal elimination happened. Number four, you will collect the embryo. This process is called as embryo rescue. For embryo rescue, by using a forceps, we will remove these weed seeds which are in green color from these spikelets, and then we will give a small cut. And after this small cut by a blade inside a laminar flow hood, by using a forceps, we will collect this small embryo. This small embryo is called as immature embryo, and whole process of removing this embryo by giving a cut and getting this embryo and shifting it to the growth media is called as embryo rescue. What is it? Embryo rescue. So the embryo rescue is done. The embryo is shifted onto the growth media, and if we give appropriate rooting and shooting media, as you can see in figure D, you will get plants. These plants will be haploid. So we have already talked. If you culture a pollen grain because it is haploid, so the plants that you will get will be haploid. So here again, because the 10 chromosomes of the maize were lost, they were completely eliminated. So there were only 21 chromosomes present in the embryo. So when this embryo is grown on the growth culture media, so embryo has one part that grows into a shoot and another part that grows into a root. So you will get roots and shoots on an appropriate rooting and shooting media. I mean, if you give auxin and cytokinins in an appropriate amount, so these plants will grow. Each and every cell of these plants, the roots, the stem, the shoots, etc., they are haploid. And we will, by using standard procedures, we will take some cells from these plants and we will do the chromosomal counting. What is chromosomal counting? That under a specific histological chromosome, that where you can color the chromosomes with appropriate chemicals so that they become visible under the microscope. So this is not a light microscope, but a better chromosome, a compound microscope. Sorry, a compound microscope, not a light microscope. ठीक है, आम माइक्रोस्कोप नहीं होती जो लैब में पड़ी होती, थोड़ी सी पावरफुल कंपाउंड माइक्रोस्कोप होती है. Under that microscope, you can count the number of chromosomes in a cell. So what you can do, you can produce protoplast. Protoplast is a single cell, and from that single cell, you can get the chromosome. And these chromosomes, on, for example, if you count these chromosomes, these are 21 chromosomes. So this will confirm that your plants are indeed haploids. Okay. So these are 21 chromosomes. So the haploid plants, as we have discussed in a previous class, the haploid plants are very weak in nature. They will not grow very fast. They will grow slow. They will be susceptible to diseases because half the chromosomes are missing. So to avoid this and to get the seeds, because these haploid plants also do not produce any seeds. So to get the seeds from these plants, we do the colchicine treatment. So I have already told you in the above picture that we will, after, for example, in the figure D, you can see these plants are on media. So we will shift them on into pots. The pots will have peat, and the pots will also have a soil, mixture of both, and we will give them nutrients for at least 2 to 3 weeks. The plants will grow. They will establish their roots in a greenhouse, in a growth chamber, in controlled conditions. After that, we will take the plants out of the pots, and we will cut their roots except 1 inch. And after that, these plants are dipped in colchicine solution. The colchicine will hinder the spindle fiber formation. So the chromosomes, the 21 chromosomes, in meiosis, there is a step, or even in mitosis. Okay, forget the meiosis. Here only mitosis is happening. So in mitosis, there is a step when the chromosome number is doubled. And after that, those, how chromosome number is doubled? Not actually the number, but you can say the chromatids. I have drawn a picture to show it to you that the chromatids will double. If there are 2 chromatids on a chromosome, they will become 4. And if we apply the colchicine, these chromatids will not be pulled to the opposite direction because spindle fiber formation has been hindered by colchicine. So all the 42 chromosomes now, they will move to one pore, they will move to one cell, and the chromosome number will double. And then these plants are again shifted into a mixture of a soil and peat for hardening purpose. And once it is done, in after 2 to 3 weeks, 3 to 4 weeks, once the plants have started growing, once the plants have uh, uh, covered, have recovered from the shock of colchicine, we will shift them to the field. And before that, or after shifting them to the field, we will again isolate the protoplast from these plants. And taking a single protoplast means a single cell. We will again do the counting of the chromosome like in figure F. So this will confirm that there are 42 chromosomes. If there are still 21 chromosomes, it means that your colchicine treatment has failed. So in field, after that, you have already seen in the above figure that bee plants, once they are shifted to the field, they will produce the seeds, and these seeds can be used for the further experiments in future. So this is the haploid production in wheat by two methods, that is microspore culture and weed into maize crossing systems.